My name is Thomas Wood and my company is Fast Data Science. I'm going to talk to you about using natural language processing to predict the risk and the cost of running a clinical trial. Throughout the drug development process, there are a number of points where there's potential for natural language processing to make a huge difference. Right at the beginning of the process during drug discovery, you have documents floating around which drug is suspected to act on which target and so on, and that's often very unstructured. And then there's scientific literature which can be analyzed. Then, during the clinical stages, which are phases 1 to 4, when the drug gets tested on humans, there are clinical trial protocols which describe how a trial is going to be run, and there are narrative reports which are written after a trial is run, as well as other types of documents. I'm doing a bit of work on all of these, but mostly on the clinical trial protocols. After a drug is on the market, you have real-world evidence. Pharma companies will interview doctors, transcribe interviews, and get the doctor's opinions on which drug is suspected to interact with which other drug, or have which adverse effect, or implicated in which adverse event, and all kinds of things like that. And that ends up generating a huge body of text to be analyzed. And there's so much potential for natural language processing with and without generative AI. At the moment, I'm working on uninformative clinical trials. An uninformative clinical trial is a trial which does not benefit the scientific body of knowledge. It does not result in a policy change. It doesn't deliver meaningful results. It's both a waste of money and a violation of ethics. There's a paper called Harms from Uninformative Clinical Trials by Zarin et al., where the authors have looked at a number of clinical trials and judged them to be informative or uninformative based on criteria such as: was the trial cited in sub-studies, and did it result in a policy change? And they have identified key factors which led to uninformativeness. What happens to a study that ends uninformatively? So a study that ends uninformatively may never finish because the sample size was too small. Maybe the investigators really needed to recruit 200 people and they only recruited 100, so they don't have the statistical power to deliver any meaningful conclusion. Maybe they didn't plan their statistical analysis correctly. Maybe it changed focus, or it was published too late, or it was never really published. There are all kinds of reasons why a trial could end uninformatively and effects that come from that. The key to a lot of this is in the clinical trial protocol. A clinical trial protocol is a document written often in very scientific English. Here's an example of one of them. You can find a lot of them online. It's quite a long document. It's very technical, and it describes how a clinical trial will be conducted. It has information about the objectives of the trial, the design of the trial, how it's going to be run, how the statistical analysis will be done, and if you want to guess how much the trial is going to cost, the first place to look would be the protocol. If you want to assess the risk level of a trial, then the protocol is a good place to look as well. Here are some interesting parts of the protocol that we often look at. The statistical analysis plan gives information around what kind of statistical tests will be done. Another one is the schedule of events. So this is a big table. It has time on the x-axis, so it says what will happen on a particular date. Then it has the action that will be undertaken on the y-axis. So if you wanted to work out the cost of a trial, you'd be interested in working out how many activities have to be undertaken, and a good proxy for that is looking at the size of this table. So I've been working on a tool called the Clinical Trial Risk Tool, and this is a software tool which allows you to predict the risk of a clinical trial ending uninformatively. The tool works by letting you drag and drop a PDF into a user interface, and it will analyze the content of the protocol using natural language processing. So here's a demo of this tool. It's online. It's free for anyone to use. You can find the source code as well. You can drag and drop a PDF in there, and you can see a number of properties of that protocol. You can also select protocols from the list that's already there. So this one has been identified as high risk. The tool has identified that it didn't have a statistical analysis plan. You can get an explanation from the tool here. So it did have an effect estimate, and the size of the effect estimate has been identified, and so on. The tool was originally designed to extract information from HIV and TB trials, and now we're expanding to other pathologies, and it's been funded by the Gates Foundation. And the target users could be an investigator who wants to get funding for a trial and wants to submit it to a sponsor and stress test it before it gets to the sponsor. It could be the trial sponsor themselves. it could be the CRO. The free version, which is online at the moment, is only doing clinical trial risk, but we're now moving on to cost modeling, which has become a little bit more complex. So how do we predict things like risks, costs, and duration? There are lots of very sophisticated machine learning models such as generative AI and very simple ones like linear regression. But where I find that a lot of value comes is if you have in-house data, such as the finance data that the pharma company would have internally. For example, data on 100 clinical trials that have been run in the past within your organization, and the number of things you knew about each trial before it ran. Quite often, a simple model such as linear regression can be very powerful when combined with this data. Imagine you know the country each trial was run in, the disease area, the number of subjects, and so on. How would you predict the cost? One very simple solution is you would just average. You would bin your past trials into classes, you would average by class, you would maybe take the average per subject by class. And that's quite simple to do. There is some public data available on past trials. For example, if there has been a pharma company involved in some kind of litigation or freedom of information request, people have collected data on these trials online. This is some of the information I've been using. One challenge that you have is that pharma companies don't often voluntarily make their trial cost data public, and you have to join it from disparate sources. So there's a bit of data mining to do to get any training data for what we're doing. I found there's a lot of advantages in choosing fewer features rather than more because of this sparsity in the number of training data examples that you have. Here are some of the features that we've identified as contributing to uninformativeness. It was quite tricky to get this list, and we used a survey method inside the Gates Foundation to identify these, as uninformativeness is a bit subjective and data is very hard to find. Overall, it's pretty widely accepted, so everyone seemed to agree that the statistical analysis plan is really the strongest feature that predicts the risk of uninformativeness. So a trial without a statistical analysis plan or with an inadequate statistical analysis plan is very high risk, because it could lead to underpowering. Perhaps the investigators have not fully worked out what sample size they need or how they're going to do their statistical analysis. And then there are things like the effect estimate, which is closely related to that. So people should really have an idea of the effect estimate that they expect to see. So we've worked out a few of these factors and we've put them into our tool. And at the moment, the free online tool uses these 8 features: pathology, phase, statistical analysis plan, uh, effect estimate, number of subject, number of arms, the number of countries, and the simulation. You can take a particular pathology and you can work out a regression model for the cost per participant or the cost of that trial. We've done that as well, and we've done things like taking factors of all these different pathologies and phases and types of intervention, and we've added those into a regression model. And we're now working on a newer version of the tool, which is also available online. This newer version will let you upload a protocol, and it will analyze it, and it will identify more features, and it will also attempt to identify the cost. At the moment, the cost feature isn't working yet. The risk is kind of working in the new version, so it's very much a work in progress. So we haven't publicized this one yet. We're extending to 30 features, and we're hoping that the user can model the cost using their own in-house cost model. We're going to allow support for multiple documents, and I'm interested also in finding out other uses of this technology, such as the duration of the enrollment period. If it is possible to identify things like that, then we could use this tool to connect the protocol, maybe to integrate with systems that the CRO would use for enrolling participants, so for recruitment. At the moment, this is all a work in progress, but it's really exciting what we'll be able to achieve once we get the entire tool working. Thank you for listening.